Good day, everybody. For this lecture video, we are going to have a review on nucleic acids and proteins. So, by the end of this lecture, we are expected to be able to discuss the different biological roles of nucleic acids in the cell, illustrate the DNA double helix, explicate how the different properties of DNA favorably contribute to its viability as a genetic material, explain how RNA structure differs from that of DNA, describe the roles of each type of RNA, and describe the relationship between protein structure and function. And for the topic outline, first we are going to discuss the biological roles of nucleic acids, followed by the DNA structure, properties, and function, the principal types of RNA and their structures and functions, then the structure and functions of proteins. Okay, so your nucleic acids are biopolymers, or they are large biomolecules. By definition, biopolymers are polymers produced from natural sources, and this can either be chemically synthesized from biological materials or biosynthesized by living organisms. And these are made up of monomeric units bonded together by covalent bonds. And these monomeric units form larger molecules, hence large biomolecules, right? And then your biopolymers are actually degradable. They find use in various industries, ranging from food industries to manufacturing, packaging, and biomedical engineering. They are promising materials owing to their characteristics like abundance, biocompatibility, and unique properties like non-toxicity and others. According to furthermore, your biopolymers are classified according to various scales. First is it is first based classified based on its origin. So it is either a natural biopolymer, which includes either a protein or a polysaccharide biopolymer. So pag protein, we have the collagen, the fibrinogen, and the soy protein. Those are your biopolymers, natural biopolymers. And for the polysaccharide, we have your cellulose, your chitin, and your starch. Aside from natural biopolymer, we also have synthetic biopolymer. This is either a biomass based or a petroleum based. And lastly, classification or basis on origin is the microbial biopolymers. So it is either polyesters or carbohydrates. Another classification is based on type. So it is either sugar-based, starch-based, cellulose-based, and synthetic-based. And the last classification is based on the monomeric units. So we have the polysaccharide, the proteins, and the polynucleotides, to which um, your DNA and RNA belong. So they are made up of monomers, which are known as nucleotides. So it was first found in the nucleus of the cell, and later found outside, found also outside the nucleus. Okay, so the biological role of your DNA no, primarily is the storage of genetic information. And this information is based on the order or the sequences on nucleotides in the nucleic acid polymer. So this is what makes us unique individuals. Each of us has a different order of sequence embedded or integrated in our genetic makeup. Aside from the storage of genetic information, it is also for determining genetic coding. No, it's for coding, which is because each of us has a different genetic codes, and then also directing protein synthesis and directly responsible for metabolic activities, evolution, heredity, and differentiation. and it is also a more stable molecule in comparison to RNA, and it holds more complex information for longer periods of time. On the other hand, your RNA is synthesized by DNA for the transport of genetic information to the protein building apparatus in the cell. It is also responsible for the synthesis of new proteins using the genetic information it has transported. And the mRNA, or the messenger RNA, transfers genetic information through the plasma membrane. So, um, as we move forward in the succeeding slides, we will going to also know the functions of the different types of RNAs present in our body. Okay, so just a little bit history. The people that you see here in the slide was were actually not the first one, or not the individuals who really discovered DNA. So it was long before them that the DNA was discovered. So it was discovered by Johann Friedrich Miescher in 1869, and in 1953 is where these individuals improved that discovery. So Micher had isolated white blood cells out of seepage collected from discarded surgical bandages. He found that he could extract a viscous substance from the cells in this material. And so, Micher also observed that most of the non-nuclear cell components could be lysed away with diluted hydrochloric acid, leaving the nuclei intact. And then the addition of the extract of pig stomach, or a pepsin, to be specific, resulted in somewhat shrunken but clean preparation of the nuclei. So it was in 1869 where a DNA extraction was first performed. And then in 1953, going back in this year, it was James Watson and Francis Crick, a Nobel laureates, who first described the structure of the DNA, and their basis on the structure was based on the previous observations of the chemical nature of DNA and physical evidence, including X-ray diffraction analysis by Rosalind Franklin. So the composition of DNA was known, and um, normally it is composed of four different nucleotides, and each nucleotide is composed of a five carbon sugar, a phosphate group, a nitrogen base, and also a hydrogen, and um well, that three, no, we have the phosphate group, deoxyribose sugar, and nitrogen base. But if you are going to expand it, we have five components of your nucleic acids. Of course, a phosphate group, a carbon, a nitrogen, an oxygen, and a hydrogen. And then, as you can observe um, in the illustration in the slide, that the nitrogen bases in your DNA, we have four. No, we have the adenine and the guanine, which are the purines. So the purines are the um, two-ringed heterocyclic aromatic organic compounds. And then you also have there your thymine and cytosine, your pyrimidines, which are the single-ringed heterocyclic aromatic organic compounds. And then, um, following the Chargaff's rule, it is stated that the number of thymine and cytosine is equals to the number of adenine and guanine present in the structure. And so, thymine, the number of thymine is equal to the number of adenine, and the number of cytosine is equal to the number of guanine in the structure. So it was formulated by Erwin Chargaff. And then, uh, as I've said, that by X-ray diffraction of Franklin and Wilkins, they suggested that the molecule was long and skinny, it also had two parallel components and was helical. And that proposition or theory was solved or was described by Watson and Crick. So is there a difference between a nucleoside and a nucleotide, sir? Well, the answer is yes. No. Nucleoside is composed only of a nitrogen base and a sugar. For DNA, of course, since there is a removal of a hydroxyl group at the second carbon, so we termed it as the deoxyribose sugar. For RNA naman is since complete na lahat ng carbon atoms na present in the sugar, in the ribose sugar, so it was not deoxified, no? So kompleto siya. So tinawag pa rin siyang ribose sugar. And then, of course, they differ on the attachment of the nitrogen base. So, yung dalawa na yan, pag magsama yan, we call that a nucleoside. Now, by the addition of your phosphate group, it will become now a nucleotide. So, yung base natin is either purine or pyrimidine. As I've said, pag purine, these are double-ringed structure. Double-ringed because meron pang isang ring dito. A purine is, as I've said, an aromatic compound, a double-ringed aromatic compound, which is composed of five carbon atoms and four nitrogen atoms. On the other hand, your pyrimidine na organic compound is composed of four nitrogen atoms, carbon atoms rather, and two nitrogen atoms. And then that nitrogen base is attached to the first it is attached to the first carbon atom of the ribose sugar. And then on the terminal carbon of the ribose sugar or the deoxyribose sugar is the attachment of the phosphate group. So, yon. Okay, so again, pag nucleoside, it is the base and the sugar. And pag nucleotide naman, it is the base, the sugar, and the phosphate group. Next, ito na, pinangunahan ko na pala yung sarili ko. So, of course, as I've said, your pyrimidine is a single-ringed heterocyclic aromatic compound, organic compound, and your purine is a double-ringed heterocyclic aromatic organic compound. And then, your pyrimidines is we have the example of your pyrimidine. It is found in your DNA. We have the cytosine and the thymine. And the thymine is not present in your RNA. No, it is only present in your DNA. The counterpart of the thymine in the RNA structure is your uracil. For purines naman, we have your adenine and your guanine. Okay. So the important features of the DNA structure and properties. So of course, it is a double helix, a right-handed double helix. Sir, is it all the time that the DNA structure is right-handed? Plus, hindi rin. No, I will tell you later. Then, of course, next is that the helices are anti-parallel. What do we mean by anti-parallel? Hindi sila magkapareha, or they are opposite to each other. Then each helix has a series of nucleotides held together with a phosphodiester bonds between the hydroxyl group and two adjacent sugar residues. And then the helices themselves are held together by hydrogen bond between the nitrogen bases. So the guanine pairs with cytosine, and the adenine pairs with thymine. So ang standard for itong right-handed na double helix, we call it as the B form. We have three forms of your DNA. We have the A DNA, the B DNA, and the Z DNA. So the B DNA is the hydrated form of the DNA structure. It is the standard form, which has 10.5 steps or pairs or base pairs per turn. So each turn ng helical structure na yan is composed of 10.5 base pairs. The E form naman has 11 base pairs per turn. Now, yun yung sinabi ko na both A and B DNAs are right-handed forms. On the other hand, your zDNA is the left-handed na form. And the zDNA has 12 base pairs per turn, and it is formed by stress or torsion. Tinawag siyang zDNA because the due to the stress and conformation, it will also change the salt concentration present in the structure, and hence will form a zigzag pattern. Kaya siya tinawag na zDNA. So the primary structure of DNA is the linear sequence. So it begins at the 5' phosphate end and ends or towards the 3' hydroxyl end. And the connection in each of these nucleotides are joined by the phosphodiester bond or phosphodiester linkages. The secondary structure of DNA is the double helix. So it is formed by two antiparallel polynucleotide chains that wound around the same axis. So the each polynucleotide, kasi antiparallel man sila, so ang isa is 5' to 3' and the other one is 3' to 5'. And so the interaction of the nucleotide, the nucleotide is formed together by, or is joined together by a hydrogen bond, and they are joined at the center, forming a central axis. And so the sugar phosphate chains wrap around the periphery, and then kani nga to ang mga phosphate groups here, or mga that are joined together by the phosphodiester bond. This is the backbone of your DNA structure, and they are the ones responsible in the formation of the helical structure of your DNA. So here, the DNA molecule usually depicted as an inflexible rod, but it can be bent or curved sometimes. So this theory was proved by Agnes Roy and Ramin Golestanian of the University of Oxford, which allowed the ordinary random jiggling of atoms at room temperature to stretch, bend, and twist the molecule, and they analyzed this motion to deduce the mechanical flexibility parameters of DNA. They found that these parameters depend on the molecule's length in ways that were hard to predict. Um, in terms of the length, the guys, or the flexibility of the atomic scale, actually the flexibility of the DNA depends on its length, no? So the long stretch of DNA is resistant to bending, and then the short strands are more flexible. So the stiffness depends sensitively on molecular length until this length exceeds a single turn of the double helix. So DNA is deformed when some proteins bind to it or when it gets wrapped up in chromosomes. So Agnes Roy and Ramin Golestanian used computer simulations to investigate DNA lengths ranging from a single base pair to four turns of the double helix. Ang four turns ng double helix is equivalent to 50 base pairs. So it is said that the shorter the length of the shorter the strand of the DNA, the more flexible it is. Kasi kung longer na or lengthy na ang iyahang DNA strand, it is no longer flexible. So your DNA molecule has major and minor grooves. So ano pala tong grooves, sir? Your groove is formed, the grooves are formed in the helix by the backbones. So, sir, saan ba makikita yung ating grooves? So, you can observe here, no? It is depicted here. The major groove, yung mas malaki yung kanyang indention, and then the minor groove. So, the major groove occurs where the backbones are far apart, and then the minor grooves occurs where they are close together. So, the grooves twist around the molecule on opposite sides. And um, is it paano nga natin ma-identify yung major and minor grooves natin? Yung major groove natin is wider than the minor groove. And these grooves allow proteins to bind to and recognize DNA sequences from the outside of the helix. The grooves expose the edges of each base pair located inside the helix, which allows proteins to chemically recognize specific DNA sequences. On the other hand, your minor groove is narrower of the two grooves in the DNA double helix. Um, it is so yun, no, it is narrower than the major groove. So again, major groove, yung wider ang kanyang um, space or indention with each other. And then, yung minor groove naman is uh, narrower of the two grooves in a DNA double helix. Now, stabilization forces in DNA are from the hydrogen bonding between bases because and the stacking in the stacking of the bases, which excludes water molecules, either through hydrophobic interaction and van der Waals forces. Yung hydrogen bond. Okay. So hydrogen bond is a chemical bond in which a hydrogen atom of one molecule is attracted to an electronegative atom, especially nitrogen, oxygen, or fluorine atom, usually of another molecule. So, as I've said a while ago, that hydrogen bond is the bond that join together your nitrogen bases. So you have here two hydrogen bonds between adenine and thymine pairs, and we have three hydrogen bonds between the cytosine and guanine pairs. Base stacking. These are the bases in DNA. The bases in DNA are planar and have a tendency to stack. So the forces that contributes to the stacking is through either hydrophobic interaction or van der Waals forces. So we have here the different structural forms of DNA. We have the B form, the typical form, which is a right-handed helical form. We also have the A form or aDNA, the dehydrated form, or which is still a right-handed helix, and then lastly, we have the Z form or the zDNA, which is formed when the DNA molecules have alternating G's and C's on the same strand. So to compare the three, no, we have here the type of helix, if it is either right-handed or left-handed, the helical diameter, the rise in base pair. So they are measured in nanometers or in angstrom. Um, then distance per complete turn, and then the number of base pairs per complete turn, and then the topology of the major groove and also the minor groove. So sir, question po. Um, is the base pair is the base pairing constant? Plus yes and no. Yes, they are constant, and no, there are also times na nami-mismatch siya. Like instead of adenine and thymine, nagbabind siya kay cytosine, adenine and cytosine. Instead of guanine and cytosine, nagiging guanine and thymine, or perhaps adenine and adenine. This can distort the DNA helix and disrupt the maintenance of sequence information. And due to their effects on enzyme and that metabolize the DNA, modified nucleosides have been used effectively for clinical application. So class, ang pagka-mismatch ng mga nucleosides na to is also naging beneficial siya in a way that we were able to formulate drugs. So sir, what are those examples? First example natin is the 5-bromouridine and the cytosine arabinoside. These are modified thymine and cytosine na nucleosides that are anti-cancer drugs, no? So kato sila. And then another one is azithromycin, or retrovir, cytosine 2,3-dideoxy 2-fluoro and 2,3-dideoxyinosine. These are mismatched na ano na that are used as drugs for HIV. And then we also have the acyclovir or Zovirax, which is used to drugs that which are drugs used to combat herpes simplex virus and varicella zoster. So you see, ang mga mismatch na mga nucleosides is not all the time uh, bad thing, no, because we can turn them into something novel, and perhaps can benefit the mankind, of which in this case it on the examples that I mentioned, it became drugs for certain diseases. Now let's move forward to the types of RNA structure and function. Sir, question po. Are the is the structure of the RNA constantly linear? Class, hindi po. Um, your RNA are they have your RNA has the ability to fold, and that folding will create a complex 3D shapes, which now become a hairpin loop or hairpin loops. And that hairpin loops will now have a specific function. So, sir, what is the example of a hairpin loop? Plus, as shown in your screen, we have now your a hairpin loop. At one of the examples of your hairpin loop, we have your messenger RNA or mRNA. So it is a single strand that carries messages from the DNA in the nucleus to the ribosomes in the cytosol so that it can be used to make proteins. It makes up about 5% of the total RNA composition of the cell. It also serves as a template for protein synthesis in a process called translation. So in eukaryotes, a distinct mRNA molecule is produced for each gene. And for prokaryotic cells naman, they have mRNA molecules that code for several genes. So again, messenger RNA or mRNA plays an important role in the transcription of DNA. During transcription, certain proteins called transcription factors unwind the DNA strand and allow the enzyme RNA polymerase to transcribe only a single strand of DNA. And then, when RNA polymerase transcribe the DNA into an mRNA molecule, adenine pairs with uracil and cytosine pairs with guanine. And then at the end of the transcription, mRNA is transported to the cytoplasm for the completion of protein synthesis. Now, let's move on to the tRNA or the transfer RNA. So tRNA makes up about 15% of the total cellular RNA. It has a distinct stem loop structure and functions in carrying activated amino acids to the ribosome mRNA complex during protein synthesis. And each amino acid has at least one tRNA made specifically to carry it to the ribosome. It plays an important role in the translation portion of protein synthesis. Its job specifically is to translate the message within the nucleotide sequences of mRNA into specific amino acid sequences. And these amino acid sequences are joined together to form a protein. The transfer RNA or the tRNA is shaped like a clover leaf with three hairpin loops. So kanang hairpin loops guys or class, kanang makita niyo din ha ang murag just like a hairpin, no? So tulo a clover shape na mura siya hairpin. That's why it is called as the hairpin loops. So again, it contains an amino acid attachment site on one end and a special section in the middle loop called the anticodon site. The anticodon recognizes a specific area on RNA called a codon, and then a codon consists of three continuous nucleotide bases that code for an amino acid or signal the end of translation. tRNA, along with ribosomes, read the mRNA codons and produce a polypeptide chain. Then the polypeptide chain undergoes several modifications before becoming a fully functioning protein. And next, we have the ribosomal RNA or the rRNA. It is a single strand in globular form that binds with proteins to make up ribosomes, which are then used to make proteins. It is made up of RNA. rRNA makes up about 80% of the total cellular RNA. It is the major constituent of the ribosomes, of which the cell machinery responsible for synthesizing proteins. Then the tRNA gives the ribosome its structure and also plays a catalytic role. In prokaryotes, there are three types of rRNA makeup. There are three types of rRNA that make up the ribosomes: the 23S, the 16S, and the 5S rRNA. So it is a component, no? Ang kaning rRNA is a component of cell organelles called the ribosomes. The ribosomes consist of ribosomal proteins and rRNA and are typically composed of a large subunit and a small subunit. And these subunits are synthesized in the nucleus by the nucleolus. The ribosomes contain a binding site for mRNA and two binding sites for tRNA located in the large ribosomal subunit. So during translation, a small ribosomal unit, a subunit, attaches to the mRNA molecule. And then the initiator tRNA molecule recognizes and binds to a specific codon sequence on the same mRNA molecule. So ribosomal RNA is responsible for creating the peptide bonds between the amino acids of the polypeptide chain. So when a termination codon is reached on the mRNA molecule, the translation process ends. And then the polypeptide chain is released from the tRNA molecule and the ribosome splits back into large and small subunits. We also have the non-coding RNA. So those three that we've mentioned a while ago are the coding RNAs. But we also have the non-coding RNAs. First, we have the small nuclear RNA or the snRNA. These are involved in transforming the pre-mRNA into a mature and fully functional mRNA. So they splice together the exons to form the mature mRNA. So next we have your microRNA or miRNA. So these are tiny RNA molecules, no, that constitutes for around 17 to 27 nucleotides in length. These are or these were discovered. This was discovered in Cenorhabditis elegans, a worm, and then its function, no, primary function is uh, they control gene expression by pairing with imperfectly complementary sequences in mRNAs to inhibit translation. Then we have your small RNA. So small RNA helps form the signal recognition particle that directs synthesized protein to their intracellular and extracellular destinations. So to summarize the differences between the RNA and the DNA in terms of their strands, so your RNA is single-stranded, but of course, as I've said earlier, it is not all the time single-stranded. No, there will be times that it will become a double-stranded na structure, and hence we have your mRNA, your tRNA, and other types of RNA. On the other hand, your DNA is really a double-stranded, except for certain viral DNA which are single-stranded. So naatay double-stranded DNA viruses, naputay single-stranded DNA viruses. Then, in terms naman of sugar content, the sugar content of both nucleic acids is of course ribose. But the difference to each of them is that in DNA, the hydroxyl group at the second carbon of the ribose sugar was removed or is not present. Then in terms naman of their nitrogen bases, thymine is never found in RNA except at the tRNA. So instead of thymine, the present nitrogen base there is the uracil. And then in DNA naman, thymine is always present and uracil is not present naman. So instead of uracil doon sa DNA, thymine yung present always. Then in terms of following the Chargaff's rule, um, it the DNA follows the Chargaff's rule because of the double-stranded structure, whereas sa tuang RNA, it doesn't follow the Chargaff's rule since it is technically or primarily a single-stranded structure. To continue, RNA can be easily destroyed by alkalines to cyclic diesters of mononucleotides, whereas your DNA resists alkali action due to the absence of the hydroxyl group at the second carbon position of the ribose sugar. The RNA is relatively a labile molecule that undergoes easy and spontaneous degradation. whereas the DNA is a stable molecule. The spontaneous degradation is very slow, and then the genetic information can be stored for years together without any change. Then the RNA is mainly cytoplasmic but also present in nucleus, primarily for transcription and small RNA, small nuclear RNA. DNA naman is mainly found in the nucleus, but there are also extranuclear DNA found, specifically in the mitochondria and plasmids, etc. The base content varies from 100 to 5,000 and the size is variable. DNA naman has millions of base pairs depending upon the organism. RNA have various types: mRNA, rRNA, tRNA, snRNA, and etc. Then DNA naman is always of one type and performs the function of storage and transfer of genetic information. There are no variable physiological forms of RNA that are found, and the different types of RNA do not change their forms. Ang DNA natin, as I mentioned a while ago, that it has variable forms, no? The A form, the B form, and the Z form. RNA is synthesized from DNA. So galing si RNA from DNA. However, it cannot go back. No, the RNA cannot go back to the DNA except if there is a reverse transcriptase enzyme. It also cannot duplicate except in certain viruses where it is a genomic material. Your DNA can form DNA by replication and it can also form RNA by transcription. And many copies of RNA are present per cell and there is a single copy of DNA present per cell. Now let's move on to the structure and functions of your proteins of our proteins. This has been discussed sa inyo way back in your biochemistry and even in your clinical chemistry 1 and 2 courses. So proteins are major components of all cellular systems. It is consist of one or more linear polymers called the polypeptides. They are your proteins are linear and never branched, and um, the different amino acids are linked together via the peptide bond or the peptide bonds, and then the individual amino acids within the protein are known as the residues, and then the smallest known protein is just 9 residues long, which is called the oxytocin or oxytocin, and then the largest one is over 25,000 residues, which is called the titin. Proteins are generally between 100 to 1,000 residues in length. In the absence of stabilizing forces, a minimum of 40 residues is needed to adopt a stable 3D structure in water. Protein sequence can be determined by systematically removing the amino acids one at a time from the amino acid through the Edman degradation. Now we just sequence the gene or DNA for that protein and use the genetic code to determine the amino acid sequence. Proteins account for more than 50% of the dry mass of most cells. Protein functions include structural support, transport, cellular communications, movement, and defense against foreign substances. So each amino acid has the same fundamental structure, and they differ only in the side chain designated at the R group. The carbon atom to which the amino group, carboxyl group, and side chain that are attached is the alpha carbon. A protein is biologically functional molecule that consists of one or more polypeptides. So as shown in this slide, this is the basic or the fundamental structure of your amino acid. So we have the amino group, the carboxyl group, and the R group on the side chain. Now sa side chain or the R group, dito nagkakatalo lahat. Or dito natin malalaman kung anong type of amino acid ang present, no? Because this is where the the different components are attached to the amino acid in order for us to identify the specific amino acid. And by that, so we in this figure we have here the different amino acids with their different side chains. So it could either be a non-polar side chain or hydrophobic side chain, polar side chains or the hydrophilic side chain, and also another hydrophilic or the with electrically charged na side chains. So for the non-polar side chains, we have glycine, alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, and proline. For the polar side chains, we have the serine, threonine, cysteine, tyrosine, asparagine, and glutamine. Then the electrically charged side chains, we have your aspartate or aspartic acid, glutamic acid, lysine, arginine, and histidine. So, how are peptide bonds formed? So, peptide bonds are formed by a biochemical reaction that extracts a water molecule as it joins the amino group of one amino acid to the carboxyl group of a neighboring amino acid. So the linear sequence of amino acids within a protein is considered the primary structure of the protein. So as it keeps on binding or extracting water molecule, then mag-attach si water molecule, mag-attach ang amino group to the carboxyl group, and it keeps on um, attaching itself, we will now have a linear sequence, which is now the primary na structure of our protein. So as it keeps on binding, it also is folding, no? So a functional protein consists of one or more polypeptides precisely twisted, folded, or coiled into a unique shape. Then the sequence of amino acids determines a protein's 3D structure, and then the protein's structure determines its function. With this, the bioinformatics uses computer programs to predict protein structure and function from amino acid sequences. So we have four levels of protein structure, having said those things a while ago. First is a primary structure, the linear sequence. This is followed by the secondary structure, which is consist of coils and folds in the polypeptide chain. Next is a tertiary structure, which is determined by interactions among various side chains, of which this is also where the three-dimensional pattern is observed. And finally is a quaternary structure, which results when a protein consists of multiple polypeptide chains. So we have here an example, no, of a primary structure, a transthyretin polypeptide. So the sequence of amino acids in a protein, and it is determined by inherited genetic information. Secondary structure results from hydrogen bonds between parts of the polypeptide backbone. So it includes the alpha helix and the beta pleated sheet. No, you have the alpha helix, the one, this one, the alpha helix uh, the structure and the beta pleated sheet. Then they are joined together by the hydrogen bond. So the beta strand shown as a flat arrow pointing toward the carboxyl end. Next is the tertiary structure. It is the shape of a polypeptide in three-dimensional na pattern or shape. Then quaternary structure naman results when two or more polypeptide chains form into a one macromolecule. Then, if we are to put it all together, we have the secondary structure that will continuously coil and fold and will create now the tertiary structure, and then finally we have the quaternary structure. So a change in primary structure such in a case of sickle cell disease. So a slight change in primary structure can affect a protein structure and ability to function. So the question here is, how does the primary structure change? So there are so many factors, no, but one of the things or one of the factors that can alter the primary structure of the protein is mutation. So, sickle cell disease is an inherited blood disorder, results from one single amino acid substitution in the protein hemoglobin. So, normally, or in a normal hemoglobin, is that it is made up of four polypeptide chains, two alpha subunits and two beta subunits. In the case of the sickle cell hemoglobin or hemoglobinopathy, there is a substitution at the sixth amino acid at the primary structure or the primary protein structure. So instead of glutamic acid, it will now become valine. And this will lead to the polymerization of mutant hemoglobin S, impairing erythrocyte rheology and survival. So the substitution of that particular protein structure is located at the beta subunit of the protein, hence exposing the hydrophobic region of that particular subunit. And it will lead to the sickle cell hemoglobin, right? If it will be formed up to the quaternary structure. And now, the molecules will crystallize into a fiber, and then the capacity of the protein to carry oxygen will now be reduced. And that's the end of our video lecture discussion on the review on nucleic acids and proteins. Should you have any questions or clarifications pertaining to the topic, please feel free to address that in your lecture instructor. I hope that you learned something today. Thank you very much for listening.